wearing it.
Yeah, same thing here. People are not wearing masks.
Yeah.
They're doing jogging, running, but
Yeah.
still the same.
Yeah.
Yeah,
And
so
if what they think that if we are not standing close by, we can remove our mask. That sort of thing is there, no? So
what come again? I didn't get you properly.
I'm saying that if they are sitting in a room and not very close, but still they will remove the mask, and that is the reason. I mean,
Yes, yes. In
what
a
they are,
closed room, it's
yeah,
the more chances to
yeah,
counter.
they're, yeah,
Yeah, anytime I think you should wear the mask. The only problem is people don't understand the complexity of the disease and
yeah,
they understand the outcome because
yeah,
it would have been, say, Ebola virus or it would have been a smallpox. People will not dare to step outside.
yeah,
It's a common flu and we'll get rid of it, but it's not the case.
yeah. But it's not the case. I have
Yeah,
seen many of the cases. The on the third day the condition get worse,
right.
hypoxia and everything, those symptoms. And if there's a
I mean,
comorbidity, then
yeah.
you cannot take a chance.
And even if you recover, you don't know the long-term effects of these viruses, what they are going to do.
Yeah, yeah. Because there is always a chance of reactivation of the virus.
Yes, of course. They can reactivate themselves. They can reactivate any of the genes in your body. You can go way
Yeah,
back. So, yeah. They should understand the disease very well before they do so casual nature
yeah,
and then move around.
yeah.
And it's very difficult to explain this to people. They would not try to understand. And it's difficult for everyone, right? You don't want to stay at home. You want to go outside. It's more
Yeah,
difficult staying at home, but still somehow you have to find a way out. So actually, I kept
I
one
know. Unless and
task
until,
to explain to her about what should be done staying at home. At least we did for 2 months.
yeah,
So the complete lockdown in Houston also. I didn't go to the lab
yeah.
for. So we were staying in home. We were ordering everything out from outside, home delivery. I didn't go to a grocery store for last 3 months.
Mm-hmm.
Just at home and then go to lab or go in early morning when people is not there, just a walk. That's
Yeah,
it.
yeah,
Masks on.
yeah. That's what we did. Like earlier, initially, it was like in March and mid-April, it was total lockdown in Indore
Yeah.
also. You must be knowing because of your
Yeah.
parents. And we did not get the grocery and everything. So we managed. And
Yeah.
that is the reason it did not spread during that time. But the
Right,
moment
right,
it
right.
was unlocked and people started like they have come out of the prison.
Yes. That's true.
I
But
think people
I
are
mean,
joining. All
this
the
is a good
participants are
time. You
joining.
can. Oh, really?
Yes, ma'am. Within 2 minutes, we are going to start.
Yep.
Yeah.
Principal sir is here.
Yes, principal sir is here.
Yes,
Good morning,
Dr.
sir.
Ram, you can hear me?
Hello. Yes, sir. Good morning. How are you?
Yeah, how fine? How are you?
I'm good. Good.
What about America Corona?
It's worse than India.
It is increased day by day.
Yes. It's almost 10 to 20,000 cases every day in
Yes,
Houston only.
in India also, in India, 32,000 cases increased. It is the highest position today after tomorrow.
तुम्हारे उससे डालो
So
तो मैं
really
ना बाकी कुछ पे
32,000?
डालूं
32,000 per day.
32,000. Wow. Yeah, it's too much.
It is too much. Now we are waiting for the vaccine. It is important now.
Yeah. Everyone is waiting for the
What about
vaccine.
your progress about your in America? What is
Uh-huh.
the progress of vaccine?
So Moderna started their trial phase 3. phase
Yeah.
1 had very good results. I'll show the data here today. So
Okay,
they
okay, 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 okay,
good
okay,
results
okay.
in that vaccine trial.
Yes, yes.
By
Dr.
July end,
Jyoti already told me that there
yeah,
will these lectures are related about vaccine and prevention, all these things.
yeah. I mean, I'll try to keep things on the basic level because lots of BSc and MSc students.
Yes, 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 yes.
That's why I'll try to
And
make them understand
and
how
also for
vaccine
the
works.
faculty like commerce and management faculties from our college.
Ah.
Oh, okay.
So
I didn't know this.
yeah,
Okay.
Video.
There are
all the faculties of the
multi-purpose
college
college.
are here.
In my college, we are running management, commerce, and science, and computer science and biotechnology also.
Oh. Sir, I was there in the college teaching in
In which college?
2009, I guess.
In which college?
Sir,
In
Ram has taught in our college as a visiting faculty in biotechnology department for some time.
In our
yes,
college.
yes.
Yes, sir, in
Uh,
Rock Market College in biotech
So
Stock
department.
market.
which year? Which year? 2007, 8? In my presence?
9. Yeah, yeah, you were there.
Okay, 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 okay.
Sir, he
Wow.
was there in
Welcome,
2009.
yeah, Ram. Okay,
Hello.
Yeah, I was there for like 3-4 months.
okay, okay.
So Varsha Tripaঠি's batch.
Ah, yeah, yeah, yeah.
So, principal sir.
Yes,
Sir, can we start, sir? This
yes,
today's
start.
Singh sir. Are you ready, sir?
Yes,
Dave
I'm
madam.
ready.
Dave madam.
Yes, sir, I'm ready.
Are you
I'm
ready?
We
Very
can
good
start.
morning to all participants. Welcome to you for today's webinars. So मैं दवे मैडम से वो करूँगा कि दवे मैडम प्लीज़ स्टार्ट करिए
I'll
today's topic introduction and proceed
Okay.
source person introduction. Please ma'am,
Let's
please,
proceed.
please ma'am.
A very good morning to respected principal Dr. Vasudev Mishra sir, our energetic guest speaker Dr. Ramkumar Singh, enthusiastic participants from various colleges, dear colleagues and students. With confirmed COVID-19 cases worldwide surpassing 13.7 million and still continuing to grow, scientists all over the globe are pushing forward with die-hard efforts to develop vaccine and treatments to slow the pandemic and lessen the damage caused by the disease. The great hope in the control of infectious disease is always a vaccine. But drugs and vaccine development is sometimes described as a pipeline with compounds moving from pre-laboratory development to laboratory preparations to animal testing and finally to clinical trials in people, which incidentally should also not cause any damage or side effects to people. It can take a year or more for a new vaccine and compound to go from initial discovery to the marketplace. Many compounds never even make it that far. That is why many medications being eyed as the potential treatment for COVID-19 are drugs that already exist and are commonly in use for other diseases. To throw some light on the immunological aspect and role of vaccine and drugs, we are privileged to have Dr. Ramkumar Singh as a speaker who will brief us on the current treatment modalities for COVID-19 and ongoing clinical trial. This young and energetic scientist completed his BSc from Aving Christian College, Allahabad in 2005. He did his MSc in Life Science from DAVV in 2009. After passing the GATE and NET, he grabbed the UGC CSIR, JRF and SRF fellowships and completed his PhD in Life Science from Advanced Center for Treatment, Research and Education in Cancer from Tata Memorial Center, Mumbai in 2016. During his PhD, he also trained and mentored summer and dissertation students at Tata Memorial Center. In April 2017, he moved to Texas to work as a postdoctoral research associate at MD Anderson Cancer Center, Houston, and has successfully pioneered 4 projects on multiple myeloma. He has more than 10 years of experience in cancer biology with 11 publications in peer-reviewed journals and 419 citations. He is skilled in molecular imaging and anti-cancer drug characterization. He has experience with generating therapeutic monoclonal antibodies against multiple myeloma and also expertise in flow cytometry, microscopy, and 3D spheroid culture. He also has proficiency in preclinical evaluation of anti-cancer compounds. He has received Young Investigator Award and a Travel Award at the 17th International Myeloma Workshop held in Boston in 2019. He won the Best Poster Award during an international conference on new ideas in cancer challenging dogmas in 2016. He was an invited speaker and guest of honor at a couple of national and international conferences on life science, cancer biology, and human health. We are overwhelmed and thrilled to have Dr. Ram as a speaker today. Now I request our principal sir to welcome Dr. Ram and announce to formally begin his informative talk. Thank you, everyone.
Thank you.
And over to principal sir.
Thank you, Jyoti. Thank you very much. Dr. Ram, I request to you, please start your lecture, please. Dr. Ram.
Thank you, sir. Thank you, ma'am, for the brief introduction. That was not required.
It's our pleasure.
It is
It's always a
essential.
pleasure.
It is essential, Ram. It is essential.
And actually, I'm very happy to be here because I have a strong bonding with the college cloth market,
Yes,
which
of course.
was there, and that was my first college for having a teaching experience for BSc students.
It was your
So,
first job as well.
yeah, my first job as well.
Great.
Right now, I'm doing my postdoctoral fellow here and working on multiple myeloma. But because of this COVID situation, more into learning things about how COVID-19 is going on, what was the reason, and what are the clinical trials ongoing. Because we majorly deals with the clinical trial for the oncology, like multiple myeloma, breast cancer, and other tumor types. But COVID-19 was new for me, and we had a 2 months of lockdown. So I used that time to learn about this virus stuff and how it works, how vaccines are developed. Because one of my project is also on vaccine against cancer biology.
Okay.
So today I would like to start with a pandemic, and I termed it as an unexpected detour to give a better view of inner self, because many times we roam around a common road to see the beauty of the nature, but sometimes a detour we take and we have a better view rather than the regular view we have. So the only best part which happened in this COVID-19 period was the pollution, which went significantly down, and Earth actually revived itself after a long period of time. So I would say science evolves with every mistakes and failures, as told by Dr. APJ Kalam, that don't follow the stories which are successful, they follow the which will lead to the message. So read the failure stories, you get some ideas and get success. Like if you know about the revolt of 1857, that is a Sepoy Mutiny, which was the first war of independence in India against Britishers. And that was not successful because that was not coordinated very well. That was not having a good leadership quality at that time. So if it would have started at one time at every place in India, it would have a success. So the drawback was the coordination. So we lacked coordination at that time. So we have to learn that we need a coordination. As of now, also we need a coordination. We should wear masks. We should follow the guidelines provided by WHO. We should follow the guidelines by Government of India. We should not step outside without mask. So these are things which we need to learn. And these are the learnings from the failures from our past. So if I talk about COVID-19 vaccines, so many people say, when is vaccine coming up? It's too late. What science and technology is doing for nothing. It's good for nothing. It's 2020 and still we have to wait so long. So if you look at the chart here, you will find the year we have spent for making vaccines. For like dengue vaccine, we have spent more than 39 years just to start our phase 1 clinical trial. And it's almost a century to make a virus against dengue. For Ebola, it took almost 27 years for identification and start of the trial for Ebola vaccine. And then it took almost 2019, we found that the vaccine was developed. And it's just 6 months for COVID-19 and people are so panicking. But still, it is a science and technology which has gone so far that in COVID-19 situation, within 3 months of time, we could start the phase 1 in human trial rather than the preclinical trials. So we should take this as a positive note that science and technology is really doing good, and we have to keep faith and hope. And looking at the history of the vaccines, this is the fastest and breaking record time for the first in-human trial for COVID-19 across the world. So let us first understand the basic structure of SARS-CoV-2 virus. If you can look into the small envelope, which is the outer membrane of the virus, which is denoted by E, and they have a spike protein, which is S, and the membrane protein, which is denoted by M, and nucleocapsid protein, which is a N protein. And it's the simplest structure of virus, and it's a RNA virus. So basically, there are two types of genome. One is DNA based, which is deoxyribose nucleic acid, and one is RNA, which is ribose nucleic acid. And this virus has a RNA as a genome. So what this small envelope protein does, it helps in the viral assembly and release. So there are a number of factors in this virus. So it needs to be assembled and disassembled. So small envelope protein does the viral assembly and release. And the M protein is responsible for mediating receptor and binding in the fusion. Viral assembly. And S protein is basically your receptor binding protein, which is major antigen of this virus, which is called a spike protein. And because of the structure, it is known as a spike protein because of the appearance of spike-like structure. And nucleocapsid protein regulates your regulation of RNA synthesis and possible antigen. When we say possible antigen, that means RNA can again be a possible antigen which can be targeted for therapeutic purpose along with your spike protein. So we move to the key reservoirs from where this COVID-19 virus came and what would be the mode of transmission for this coronavirus. So for sure, we know that this is SARS-CoV-2, which is 65 to 125 nanometer in diameter. Why I'm saying this? Because this is so small that definitely you cannot say it, but it's very hard to avoid this virus. So that's why we say wear mask all the time, because it can go through your nostrils and infect the people. And primarily host was considered to be a bat, but what is the intermediate host between bat and the human, it's not very clear, and how it is transferring from one person to another person other than human transmission itself. So intermediate host, we don't know yet, but there is definitely a community transfer that is leading to a huge spike in the number of cases of COVID-19 recently. And just have a brief life cycle of SARS-CoV-2 in the host cells. So SARS-CoV-2 is a virus which is giving rise to COVID-19 situation. And host cell is our own body, which is suppose epithelial cells of lungs. So what happens is at the initial stage, when you inhale these viral particles in air, when we don't have mask, of course. So what is happening is this is binding to the epithelial surface with ACE2 receptor, which is present on the alveoli. And this spike protein and of the virus binds to this. And one inside the cell. So this cell is formed as an endosome. And what is happening, this is acting as a cargo. When I say cargo, that means it is acting just as a shuttling vehicle for this virus from outside to inside host. So when it is inside the host, this fused endosome is getting cleared off, and this RNA genome of the virus is getting out. So once RNA is out inside the cell, it is trying to replicate and form its product. And what are the products? After translation, it forms all the necessary proteins which was required for formation of complete virus, which are basically envelope protein, your spike protein, your membrane protein, and nucleocapsid protein. And all of the information are encoded in your RNA genome. So this whole RNA genome knows how to make small envelope virus, envelope protein, how to make spike protein, how to make M protein, and how to make nucleocapsid protein. So basically, this RNA genome is your blueprint for all of the proteins which is required for a functional virus to infect. So you can see here that viruses are getting formed and they are getting assembled, and now they are getting released outside. So, and this is not only one virus, thousands and thousands of viruses are formed from one single cell. So there is a loads of viruses which are being formed every second. And this is just a timeline to show the first case was identified in China in December 8. And now it is July, and still the cases are shooting up at significant rate just because some people are not following the guidelines, people are not wearing masks, or they are not washing their hands with sanitizers, with soaps, and etc. So this could be the leading cause, and WHO already declared this as a public health emergency for international concern because it's a global and it's a pandemic in nature. The clinical characteristic features, for this COVID-19 is fever on admission for 43% of cases, fever during hospitalization 88%. So you can see that appearance of fever is a major clinical characteristic feature for this COVID-19, along with cough, sputum production, shortness of breath, and myalgia, sore throat, headache, nausea or vomiting, diarrhea. I mean, it could be independent of COVID-19, but these are the clinical characteristics which have been put together on the basis of 1,000 COVID-19 cases, and the percentage fraction is given here. So if considering 100%, 88% times people have fever and 3.8% time they have diarrhea along with COVID-19 infection. And these are the images of CT findings, including round glass opacities. So the best way to identify this COVID-19 infection is through X-ray. You can see the opaque structures, but it needs a very good radiologist. So if you have a definite infection in your lungs, you can be seen through X-ray film as well. So I don't want you guys to be confused here. Just take your eyes on this line. This axis represents the age and this axis represents the susceptibility. So when I say susceptibility, that means what are the chances of you getting COVID-19 infection when you come across a positive patient? Say there is a positive patient and I go and meet with him, and what are the chances of me getting that COVID-19 infection depending on my age? So if I'm say 30 years old, I would lie somewhere around here, right? So the susceptibility is say 0.025. But a person of age 50 years, the susceptibility goes to 0.05. That means with increase in age, the older age person are more susceptible to get COVID-19 infection when they come in contact with a positive patient. And similar is the case with the clinical fraction. So clinical fraction also increases with the age. So the major issue with this is the people who are at old age, the people who have certain medical conditions, they are definitely at a higher risk. So it's more advisable that people of age say 1 to 25 or 30, they should be very careful. They can be asymptomatic in nature, but they can carry the infection to their home, to their parents, to their grandparents. So just think about your parents and your grandparents, and don't go anywhere else for common people meeting or gatherings or some grocery shops. Just stay at home, wear mask all the time, because you will be putting your parents and grandparents in danger. So this is a life cycle of coronavirus again, and I kept this slide deliberately here because to show what are the points where we can attack these viruses or we can develop a therapeutic targets. First is at the point of entry. So if our borders are sealed and someone is intruding in our borders, like terrorists moving from Pakistan to India, so we would like to have a good defense mechanism at the border, right? So the first point of entry is a viral entry at the epithelium surface, which is through ACE2 receptor. So what if we block this ACE2 receptor? So if there is no ACE2 receptor, there will not be any viral entry and you won't get the infection. But nonetheless, it is happening. So another stage is the formation of endosome maturation, because there was a large noise about hydroxychloroquine, which is a malaria anti-malarial drug, which was being used for treating malaria, but now it was repurposed for COVID-19 to see if there has any good efficacy for this drug or no. So basically what it does is it stops the endosome formation. So there is no endosome formation. So viral particle, although they entered, they will not be able to survive. And other is the protease inhibition. So viral RNA, when along with the protein, it can degrade the protein here and the RNA will be destabilized. And other is inhibition of RNA genome replication. So suppose a virus enters the cell, but if it cannot replicate its genome, there is no point of getting more viruses out of it. So it has to reproduce. So virus reproduces by replicating its genome. So 1 RNA to 2 RNAs, 2 to 4, 4 to 8, 8 to 16. That's how they complete their genome, because without genome, there won't be any cell. So we can inhibit the viral replication at this stage with say remdesivir, which is an antiviral drug which inhibits replication of the genome. Now coming to drug development and phases of the clinical trial, it is very important to understand how clinical trial works and how the drugs are developed for any of the disease prevailing in the society, be it COVID-19, be it Ebola virus, be it chickenpox or smallpox. So I would try to touch up the basics only. So there are say three phases of the trial: phase 1, phase 2, and phase 3. Before that, there is a preclinical trial. So when we say preclinical trial, that means we are not in the clinic, although we are doing the trial for the drug to see the efficacy of the drug, whether it's effective or not. So this first step of is the development of new drugs using tissue culture or biotechnological methods or biochemistry techniques to use and identify the drug molecule or identify the target where we can hit that at most and stop the inhibition. Information on the mechanism of action, how that drug works, how it can inhibit the viral entry or toxicity of the drug. So say your drug is acting on the virus and it is killing the virus as well. But at the same time, if it is very toxic to the humans, it is not safe to use, right? Because you will be killed either ways, either by the virus or by the drug itself. So the preclinical trial is must to achieve the good efficacy and no toxicity with your PKPD obtained from these studies. And these preclinical trials are usually done in the mouse model. So we use lab mouse or other animals, small animals, to test these drugs first before entering in the phase 1 trial. So when we say phase 1 trial, that means this phase emphasizes safety, which involves 20 to 80 healthy volunteers, and information on the drugs for the most adverse effect. What are the most adverse effect on those human patients? And information on the mechanism of action of the drug, its metabolism and its excretion, how the drug is metabolized inside the body. So these are the basic aspects we look into the phase 1 clinical trial. So once we know that there is safety, the drug is not toxic, and there is no adverse effect, we move to phase 2 clinical trial. So when we say phase 2, the goal of phase 2 trial is to obtain a preliminary data on whether the drug works in a patient who have a certain disease. So if the patient have a disease and if that drug works on them, it typically involves the hundreds of patients. So now the patient from 20 to 80 increased to hundreds in number here. And the basic information what we are getting here is safety, which is continued from the phase 1, and short-term adverse effect of the studies. So what could be the short-term adverse effect across the patient samples? Now here, if we know that phase 2 trial, that the drug is working fine and there is no adverse effect, we move to phase 3. So in phase 3 trial typically involves thousands of patients, and information is more on the safety and efficacy, how the drug is efficacy across the broad spectrum of the patient across the given area. And once the phase 3 trial is over and we have a results which is very provocative in terms of higher efficacy and low toxicity, the company applies for FDA application to review and allow them to market those drugs, and that drug can be used in the market. So phase 3 trial is successful. The sponsors or appliers, they apply for the new drug application to FDA. And this process includes the review of the proposed professional labeling and inspection of the manufacturing, along with the FDA marketing, and phase 4 for post-marketing purposes. And phase 4 is of course post-marketing, that has nothing to do with the drug coming to the market apart from your economy and the budget issues. So overall, why I'm showing this timeline is because development of any drug takes time, and you have to be patient to see a drug coming to the market, because there are lots of studies going across the globe, be it mRNA, be it protein. People are targeting n number of things to find out the cure for this COVID-19, but still, I think it will take some time, and we cannot rush within this clinical trial. Say we cannot just go from phase 1 to phase 3. People may think that phase 1 there is no toxicity and there is some effect, we can directly go to phase 3, but no, we cannot do that, because we cannot just put our patients in danger just to get something so quick. So we have to be patient, and we have to give the proper time for each and every trial which is required to gather as much information as we can for the safety and the efficacy of the drug. So with this, I'll cite an example of remdesivir. So remdesivir is already FDA approved drug. So we don't need any phase 1 or phase 2 or phase 3 clinical trial for this because it's a approved drug. We know that this is not toxic or it does not have any adverse effect. But what we can do is we can repurpose this drug. So this remdesivir is basically an antiviral drug, and we wanted to test on the patients whether this is acting against COVID-19 or not. So what was done in this paper, and this is a New England Journal of Medicine, which is a very reputed journal for such kind of studies. So in May 22, this was published, and 1,000 patients were randomized in two halves, 520 and 538 patients. When I say randomization, that means we randomize the patient randomly, as the name suggests, because we don't want to bias ourselves about the patient who can recover. So we'll keep them in this group, or patient who is very ill, we'll keep in placebo to get good results. So basically we do the randomization where we don't know about any details and we just randomize the patients. And one group receives the placebo for 10 days. Placebo means there is no drug. This is just a saline, say, and the other half receives remdesivir for 200 milligrams on day 1 and 100 milligram on day 9. And when the comparison was made on overall recovery, it was shown that the patients which were receiving remdesivir, they had a superior overall recovery compared to the patient which did not receive remdesivir over the placebo for 10 days. So this was a very good data suggesting that remdesivir could be used for the patient with COVID-19 infection, at least to improve the recovery process. And this is just a graph comparing remdesivir with the blue line, and the red is your placebo. So the blue line is always running above the placebo. That means the efficacy of the drug or the action of the drug is better in case of the patient who received remdesivir. So the cure, so I'm sorry, not the cure, but the recovery. So recovery is more in case of remdesivir treated patients compared with the placebo in all the cases. And this is a recovery rate ratio. So just don't bother about the graphs and everything. The basic idea is to show that the remdesivir, where it performed better, was in the most of the cases compared with the placebo. So compared to placebo and remdesivir, the better recovery was in remdesivir because of its nature of the drug, which is antiviral in nature. So the conclusion will be that remdesivir was superior to placebo in shortening the time of recovery. So if placebo, people are recovering in say 28 days, with remdesivir, people will recover in say 5 days. Another drug which was already in the market was dexamethasone, and this study was done in University of Oxford, where they again repurposed this dexamethasone to use against COVID-19. So there was a recovery trial. So all trials are under one common server. You can go and Google it. The number of those clinical trials you can find online. So just go to those NCT numbers and you can find out how the clinical trial is ongoing, how many patients they are recruiting, whether they are recruiting patients or no. Every information is available on the Google, and it does not need any of the subscriptions. So this trial was named under the recovery trial, and this was a large randomized trial with possible treatments of patients admitted to hospitals with COVID-19. So under this trial, over 11,000 patients have been randomized in different treatment arms. So the one arm was lopinavir or ritonavir, which is commonly used to treat HIV, because people are unaware which drug is going to act, right? They don't know. So what they did, they just randomized all the patients, and whatever the basic knowledge they have about the disease and the current treatment modality in our shelf, they used that one or repurpose those drugs for the treatment. So they used ritonavir, they used dexamethasone, they used hydrochloroquine, they use azithromycin, which is a common antibiotic in India we use for common cold, tocilizumab, which is again anti-inflammatory treatment given by injection, and calmicin plasma therapy. So plasma therapy means one person who is recovering or who has recovered from COVID-19 infection, they have antibody produced in their own body, and their plasma can be transferred to the COVID-19 positive patient, and that antibody can take care of the disease burden. So I would like to mention that 11,000 patients have been equally divided into all these groups. So one group was receiving ritonavir, one group dexamethasone, one group SCQ, one group azithromycin, so on. So dexamethasone is a wonder molecule which inhibits your leukocyte infiltration. It is an anti-inflammatory immunosuppressive agent. It's a glucocorticoid agonist. So I kept this slide for the students who are in BSc and MSc. They can go and read about dexamethasone, how it works, how anti-inflammatory drugs work, and they are freely available over the counter. But before taking any medicine, you have to consult your doctor. But you should understand the mechanism of action, how any drug works, why there is an inflammation, and what can be done to tackle that inflammation. So dexamethasone is a wonder molecule, which is a very good anti-inflammatory molecule, which can take care of leukocyte infiltration, inflammatory responses. It suppresses your humoral immune responses, and it's an anti-inflammatory molecule. So using dexamethasone, what they did, they used 6,000 patients. And as earlier I mentioned, they randomized into one with the usual care alone and one with the dexamethasone. And these are the terminologies, you don't need to get into it. The basic idea over here is the patients who received dexamethasone 6 milligram per day, the mortality rate for 28 days were much better and significantly high compared with the usual care alone. That means the patients who were given dexamethasone, they performed much better compared with the patient who were not given dexamethasone rather than the usual care. So based on these results, it is suggested that one death would be prevented by the treatment of around 8 ventilated patients. So say in ICU, there are 8 ventilated patients. Out of 8, at least we can save one patient with the dexamethasone itself. So you don't need any vaccine for this to be developed, right? Although we need it, but right now we don't have that. So we use the drug we have in our shelf. So we can repurpose those drugs to treat the patients. Another aspects of treatment is convalescent plasma for COVID-19, as I mentioned earlier. So suppose this is a COVID-19 patient who recovered after some time by himself. And when I say he or she recovered, that means they have an immune response against that COVID-19 virus. So immune response is basically whenever you have any kind of infection, your body has an immune defense mechanism. It can raise your B cells, it can be CD4 T cells or CD8 T cells or antibodies which can take care of it. So in this case, there are antibody responses which has been generated in this patient. That means this patient has an antibody against COVID-19 virus. But we cannot replicate that antibody because we are not sure which antibody is that and which antibody is binding to that COVID-19 virus. So in a simple way, what we try to do in the clinic is we draw the blood, we do apheresis. So apheresis is a process of separating your plasma from your blood. So that apheresis plasma has antibody cohort in it, and this antibody will be passed on to the other patient which is infected with COVID-19. So it could be a prophylaxis. That means it could be a short term or it could be a therapeutic if it is a long term. So it is very helpful from a long period of time. It's not only COVID-19 that people are doing plasma therapy. Plasma therapy has been used for lots of diseases where antibodies were not found at that time. So it's the best way, but then we don't have many donors available for this plasma therapy where we can take the plasma and transfer to the diseased patients. So this is the latest work by Moderna on COVID-19 vaccine candidates. We are very hopeful with this Moderna's work, and this result was published day before yesterday. This is a very huge collaboration with the Modernas and different institutes. And this data was again published in a New England Journal of Medicine, where the candidate vaccine was mRNA 1273, which encodes for the stabilized perfusion SARS-CoV-2 spike protein. So when I said there are spike proteins on the surface of the viruses, so that spike protein can be a potent antigen. So instead of having that antigen injected in the body, what they did, they took the RNA, which is responsible for making those spike proteins. So they conducted phase 1 dose escalation with open label trial, including 45 healthy adults. So since it's a phase 1, So they used 45 patients here, and those 45 patients, they received 2 vaccinations 28 days apart with mRNA, which is messenger RNA 1273 in a dose of 25 microgram, 100 microgram, or 250 microgram. And what was the findings? The mRNA 1273 vaccine induced anti-SARS-CoV-2 immune response in all the participants. And this was a major throwback because uh, we won't expect that all the patients will respond to those vaccines. But in this case, what they have shown is all of the 45 have the antibodies similar to the antibodies which was found in the patient which had already recovered from COVID-19. So no trial limiting safety concern were identified. Since it is a phase 1, we are also concerned about the safeties. So there was no safety concern identified in phase 1. So that was the best news so far in last couple of months. And now around 30,000 adults of both the sexes will be recruited for the study, and which should run for approximately 3 months. And participants will receive 1 intramuscular injection of 100 micrograms of mRNA or a placebo on the other arm for day 1 and on day 29. And the trial will be conducted across 87 locations throughout US. Right now it is only in the US. Once we know that this is working, it could be globalized. So how does it works? So mRNA for spike protein of virus. So this is the RNA, and this RNA will be injected in your muscles, and this RNA will form a spike protein. So just like transcription and translation. So this is after the translation process, this RNA will form the spike protein. So instead of injecting a spike protein directly here, what we are doing is we are injecting mRNA that is forming the spike protein. So as the spike protein forms inside the body, it's a foreign body to our immune system that will recognize that spike protein, and the antibodies will be generated against spike protein only. And that's how it is going to trigger the immune response. And once the active virus is going to infect, we already have an antibody against spike protein, so that will not be affecting the healthy individual, which is already immunized with your spike protein. So this is the spike protein, and if it is a viral protein which is getting entered inside, the whole virus is getting inside. But what this group is doing, they are just taking the sequence of RNA which is forming your spike protein. As I mentioned earlier, this is the whole genome which has a capacity to form each and every part of this virus, be it membrane protein, spike protein, or anything else. So what they are doing is they are taking the gene which is responsible for making spike protein. So the only spike protein will be injected in your body, and this spike protein, immune response against spike protein, will be generated, and that's how this antibody is going to tackle your viral infection in the host. So that was all for today's talk, and this is the last slide, and I would like to emphasize more here because uh, I heard about people getting mad and people saying that I cannot go outside, people getting depressed. Lots of people have mental issues. They are not trying to retain inside the house, especially kids and youngsters. So what I would suggest is this is a good time of the lockdown. You can utilize this, read some good books. Don't follow your social media literature because day to day WhatsApp messages, Facebook messages, they are crowded with everything. Everyone will come and say, use this one, use this drug, use that drug. So don't follow that. Read good books, read good immunology books for BSc and MSc students. You can go to the library, get some good books from the institute and read the book. At least you should know how this virus thing works. What is the replication? There are n number of things to study. This is just a gist what I presented today. There are different kinds of viruses. There are DNA viruses, there are RNA viruses, there are double stranded DNA viruses, there are single stranded RNA viruses, there's a positive strand, negative strand. There are lots of things to study. So if you start reading good books, you'll get a good knowledge rather than listening to me or listening to somebody else. Try to learn by yourself. You'll have a number of questions. You can anytime shoot me an email with your queries. I'm happy to answer those. But just go on and read some good books. Explore yourself. Hobbies like singing, dancing, painting, poetry, reading, writing. Explore yourself. Search your within what you like most, because now you don't have to go to your college. You don't have to go to your work. You don't have to do anything. You just sit down and do whatever you want, whatever you like, right? And there are lots of free online courses. I myself did 2 or 3 courses online through Coursera and other online courses. So you can just go online. They are all freely available. You can enroll and you can learn new techniques. If it's a bioinformatics technique, you can learn Python, you can learn R, you can learn any language free of cost. And there are good teachers online, and you'll get a certificate too. And sometimes unexpected detour gives a better view. So my idea is try to overcome this adversity with a better plan and come up with new thing which you try to explore yourself, try to prepare yourself once this is over, what you would like to do next in your career and anywhere else. Just stay healthy. It's going to be okay. And thank you. And I'm ready to take your questions. Thank you. I hope I didn't take much time. I cannot hear anyone.
Hello.
Yeah, I can hear you.
Yes, sir. Sir, can we go forward for the question hour session?
Yeah, sure.
Yeah, sir. Is there any participant who want to ask some questions?
Hello. Anybody? Any participant for the question?
Yes, sir. Good morning, sir.
Good morning.
Good morning.
Yes, go
I'm
ahead.
Sonali Karnik, Professor Sonali Karnik from Cloth Market Institute of Professional Studies. Sir, you told about plasma therapy. So, if any person can give blood, or healthy person, or any age group of person can give blood for plasma therapy?
No, only person who has encountered COVID-19 infection and they have recovered. So
Okay.
plasma therapy, basically we are looking for the antibodies against the infection. So since the person has encountered and recovered, that means his or her body has formed antibodies against that. So basically what we're doing is transferring antibodies from a recovered patient to a diseased patient.
Okay, sir. Sir, one more question. You told that it is spreading by air also. But some studies and reports are saying that it is not spreading by air and only by swab or droplets of people. So, what is what is the true thing, that it is spreading by air or not?
So see, there is a misconception when we say airborne disease. When we say
Okay.
airborne disease, that means your disease can travel at much distance, right? From say 200 meters or a kilometer or even more than that, airborne diseases. But when we say airborne disease, airborne COVID-19 infection, that means if you are standing within 6 feet distance and someone is sneezing or coughing and is breathing fast, so those air droplets are entrapped in the air and it can travel through certain distance. So that's how we call it as a air transmit through air, just not in direct contact.
Okay, sir. So it means because we are keeping distance, then it will be less possibility of suffering from COVID.
Of course, and with mask too, because if you don't have mask, you are more likely to cause the infection. So basically, there is one more interesting study in US. So what they did, they took agar plates
Okay.
and at distance, different distance, say 6 feet, 3 feet, 2 feet, and 1 feet. At distance, they wear the mask and they did not wear a mask. and they try to cough, and they use those agar plates to culture and look for the microbes. So
Okay.
it that at a 6 feet distance, if you have a mask, you don't have any growth on the agar plate, and if you don't have any mask, it is full of the microbes. That means forget about COVID-19. It's a bacteria which can travel for 6 feet distance, and virus is a small particle, and it can be in the air suspension for a longer period of time.
Okay, sir. Sir, one more question. Which type of mask we should use? Because there are so many types of masks are coming in market, and we are confused that N95 is there, cloth masks are there, surgical masks are there. So, will you please guide which mask and hand gloves we should use, which will not, which will save us from this?
See, N95 masks are not available everywhere, but at least you can wear a cloth mask. That should be sufficient, because it will at least stop your big droplets I mean, water droplets or big um, droplets which is being transferred from one person to another person.
Okay. One more question, sir. There are no symptoms nowadays of COVID-19, and people are getting COVID positive. So, what is the reason that we are not getting symptoms and we are suffering from COVID?
So when you are saying you are asymptomatic, you are not suffering from COVID, right? You don't have any symptoms because your immune system so everyone's immune system is not similar to each other. So that's the reason some people are recovering towards COVID-19 infection and some people cannot recover. They are going to die, right? So it's about the immune system of an individual person.
See, Dr. Srivastav here.
Okay, thank
I
you very
want
much,
to
sir.
know
Thank you.
that for how long these antibodies works, and that person won't catch COVID-19 again, who has developed antibodies, and for how long it will work?
The timeline is not very sure how long it's going to work. There might be a secondary infection, but that will not be as harsh as the first infection. So there are most unlikely chances that the second infection will come up if it is a COVID-19, other than any other secondary medical conditions.
Or we can say that if antibodies develop in the body, they can recognize easily at the second time.
Yes.
To the virus, isn't it?
Yes.
So they may
Yeah.
work more efficiently.
Yes.
Okay.
So there are high chances that he will not get or she will not get the COVID-19 infection next time, but it is not for sure. It's not like you are immune or you have antivirus antibody in your system, then you can roam freely.
May or may not be.
You might be a carrier again, right? You can carry the disease and you can transmit to others rather than getting yourself.
Okay, Dr. Srivastav here. Thank you.
Thank you so much.
Anyone else for the question? Participants, anyone
Yes.
else?
Hello, sir. Sir, this is Deepmala Pathak from Sri Cloth Market College. Good morning, sir. Sir, I want to know what is the meaning of this second wave of corona, when everybody like is already maybe most of the people may be having antibodies or whatsoever. So what is the meaning of this second wave? Will it be more severe, or what is the meaning of this?
So when you say second wave, that means there was a lockdown because of the first wave, right? So when you did the lockdown and you did not see any infection at that time, and when the lockdown was opened, again it is surging towards the infection. So that is known as the second wave. So it will not stop until you maintain a 6 feet distance or you wear a mask.
Sir, but in a country like India, how is it possible to, like, if we talk about Bombay, Dharavi, and there are many places, like in Indore also, it's not possible to have that kind of a social distance.
No, it is why not? It is possible. Say if it was Ebola virus or it was a smallpox, would you dare to go step out of your house? I would say no.
So do you think it is like comprehensive to all the people irrespective of their educational qualification or their state like financial status also, and the all the things in all the conditions in which they are living? Is it possible for everybody to comprehend it in a similar manner?
See, I would not take this point because if you say literacy or people awareness, see, all people are aware of this disease, irrespective of their financial state, irrespective of their economical background. But the adversity of the disease people are unaware of because they think that this is just a cold flu, and there are lots of people who are recovering, and I would recover again. So
Yeah, exactly, sir.
take the
Because
risk.
yeah, a myth is also going on that all many things they are saying that turmeric milk or many other things will be able to defend you from this disease.
So
So
lots of
there is
people
no
think
need
that
to.
you will have a kadha every day and then you can go out, right? Definitely that might work to protect the infection. When we say you should do a gargle every day, you should take a Vicks vapor in the boiling water and take a deep breath. So these things are making you less vulnerable, but they are not making you immune. So you can do gargle every day with the salt water. That will be helpful for your throat. You cannot get infection because you are not prone to any other infection, right? So your microenvironment in your throat is more vulnerable or it is less vulnerable, depends on the daily routine activities you do. So
So
that might
may
certain extent but not making you immune.
Okay, sir. So may I ask you if you can suggest if there is some difference in political attitude as well as means attitude of both Janta and Neta in America and India. So can we learn something from them or any suggestions like?
See, uh, what I believe is it's nothing to do with the politics or it has nothing to do with your neta or your police. It is self-policing, I would say. You have to constrain yourself inside your house because it's your family and it is you whose life is responsible for everyone, right? Say if I am living, my parents are in Indore and my brother is there, my nephew is there, my niece is there, but no one is going out, right? My dad has a diabetic condition, my mom has certain medical conditions, so they are more prone and they are more vulnerable. So I would not dare any one of my house person to go out for anything. So you can fast for 2 days or 3 days. It's better rather than going outside in a situation where there is more chances of having COVID-19 infection. So what, no matter what politicians are saying, no matter what people are saying, that this is okay, you can go out. Now we have take off the COVID-19 lockdown. Now you can go. But still, I would say not to go because it is for the emergency services or urgent services that people can go out to take some medicines. People who are on diabetic medicines, they need some diabetic medicines, right? So they need to go to the pharmas. So let them go to the pharma rather than you going there and just doing gathering and roaming around with friends. So minimize the personal interaction with any of the person outside your house.
Okay, so thanks a lot. I must say that your session was highly enlightening, and we are very lucky to have you.
Thank you so much for having me.
Doctors are taking too much care, even they are pre-medicated. Still, they get few of them get corona. So, what is the lacuna remains, or what are the reasons? So they get it, and how can it avoid?
Sorry. Can you come again? I didn't hear you properly.
Yes. Okay. I'm asking that doctors are taking too much care. Even they pre-medicated also. Still, a few of them are getting corona. So what lacuna remains, or what are the reasons that they are taking? They should not do. They should take precaution for that. Because my daughter is doctor and she's working in Mumbai hospital in corona ward. So I want to know. I always remain worried about her.
See, I would explain in this way that hospital is a closed system.
Yes.
In open air, suppose 10 people are moving with the COVID-19 infection, but there is a lot of space for virus to settle down on dust, on earth, on everywhere, right? But in hospitals, what is happening is there is a rush of too many patients in there. So the whole air is contaminated for COVID-19 virus because all the people are coughing and
I couldn't
sneezing.
hear you, sir. Please again tell me.
So what I'm saying is hospital being a closed system, the
Yes.
concentration or the titer of the virus inside the hospital is significantly high compared to the outside environment.
Definitely.
And the doctors, when they are seeing the patient, although they are maintaining everything, but still it is a virus. You never know from where it can come, right? You have a PPE kit, but maybe it's not sufficient enough. You are doing something which is not helping you out. You are sometimes, I mean, out of, I mean, it's a innate or involuntary action of your muscles, because many times we keep our eyeglasses like this, or we scratch our head, or we touch our face unknowingly, and we don't recognize that we are doing itself, right? It is an involuntary motion of our hands. So many times we do that, and
Been covered, sir. Nothing remain open. Everything is covered there.
no, but at some time you are taking out your
Yes,
mask.
yes. During that time.
Yes. So say I'm wearing my apron, I'm wearing my head guard, and I'm wearing my mask. But when I'm going out and dressing room, I'm changing clothes. I'm taking it out, right? So when I'm taking it out, I'm touching my the external surface of the gown
Yes.
and external surface of the mask. So there is a way when we how to wear the mask, how to take out the mask. You cannot take out the mask from holding it here, right? Because the virus may have on the outer surface of the mask, and when you touch it with your hand, you might have those viruses on your hands. So I would say that viruses are so small that they just need a small space or small moment where they can get through the infection. But compared to the normal public outside the hospital, and the doctors are having less positive cases compared with the patient, and the
Yes.
amount of patient they have in the hospital.
Okay, okay. Very insightful lecture, sir. Congratulations to Clock Market College also. Thank you so much.
Any further questions? Any further questions from the participant?
अदिति मैं जो चर्चा चाहता हूँ अक्सर से
सर मेरी आवाज़
ये समझ आ रही है बेटा
मेघा मैम, हेलो
Hello. Anyone else for the question?
Yes, sir. I need to ask. Hello.
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 Yes.
हाँ मैगम मैम.
हाँ जी सर.
हाँ क्वेश्चन मैम.
हाँ ओके. सर मेरा ये क्वेश्चन था कि जैसे अभी फाइनल ईयर के जो स्टूडेंट्स हैं जिन्होंने एग्ज़ाम दे दिया एंड दे आर सीकिंग फॉर दी जॉब्स, जैसे आपने लास्ट में कहा कि हमें बाहर नहीं निकलना है, तो अब उनके दिमाग में भी ये टेंशन है कि टिल व्हाट टाइम, लाइक कितना समय लगेगा इस सिचुएशन को ख़त्म होने में, और कब वो जॉब ज्वाइन कर सकते हैं या फिर अपना करियर आगे देख सकते हैं. तो उनके लिए कोई मोटिवेटिंग या फिर कोई ऐसी चीज़ जो उनको घर में भी एंगेज करके रखे, कुछ ऐसे पॉइंट्स अगर आप डिस्कस करते हैं.
मैम मुझे लगता है कि अगर वो काम करना चाहते हैं या जॉब करना चाहते हैं, दे नो विच फील्ड दे आर इन, आई वुड सजेस्ट दे शुड गो ऑनलाइन एंड take trainings. Online trainings are there, online trainings which are free, and they can increase their CV and they can match up their CV to a higher extent so that they can apply to the next job, which is having much more lucrative offers, and they can have a better position. So this 2-3 months or 4 months or 5 months sitting at home, they can plan where they want to apply. They can target, say, I want to apply a biocon company for say scientist position 1. And now, since there is time for 3 months or 4 months, I would try to evolve my CV in such a way that I can apply for scientist position 2. So if they need, say, certain expertise, cell culture techniques, or if they have a bioinformatics techniques, then you can move a ladder up. So what I would do is I would go online and search for Coursera or some online courses where I can learn more bioinformatics. So right now I don't know bioinformatics. I am the student of biochemistry, and I have no clue how bioinformatics work, how search engine works, how we can do a high throughput analysis. But for these 2-3 months, I can get into the online, and I can look for bioinformatics courses. I can learn some language like Python and C language or C++ language, and these are all are freely available online. You can just go online and see what courses you like, and just do those courses, add in your CV, and once it is opening, then you will be much ahead, and then the other people who are just sitting down and complaining about the system and complaining about the people around, that this not gonna work, people are gonna die and all this stuff. So best part is take it positively, do some courses, evolve your CV, do some good work. That's it.
Okay, thank you, sir. Thank you so much.
Anyone else? Anyone else?
I want to ask Dr. Ram, this is Jyoti here.
Yes, ma'am.
Do we have any evidences as to how long this coronavirus survives in water?
No, ma'am. I don't know of this yet, because in water, I'm not sure what is the time frame. I'll look for it. If I come across, I'll get back to you.
Yeah, you do. Please share with me.
Sure, sure.
Anyone else for the question? Principal sir, can we conclude this session?
इसके पहले कंक्लूड के पहले 2 मिनट डॉक्टर राम से चर्चा करें.
जी.
डॉक्टर राम, कैन यू हियर मी?
Yes, sir.
यस. फर्स्ट 2 इम्पोर्टेंट चीज़ है जो यूनिवर्स के अंदर सब ये पूछना चाहते हैं. पहला प्रश्न तो मेरा आपसे ये है कि वो शुभ दिन कब आएगा जब कोरोना वैक्सीन आएगी. आपके हिसाब से आपका सोचते हैं कि कोरोना वैक्सीन इंडिया में या वर्ल्ड में आए, ये एक यूनिवर्सल क्वेश्चन मैं आपसे पूछ रहा हूँ. ये सब चाहते हैं कि कोरोना वैक्सीन कब आए. तो दूसरा मेरा एक कहना है कि अमेरिका के डेथ रेट और इंडिया की डेथ रेट में जमीन आसमान का फर्क है. मेरा ऐसा मानना है कि इंडिया के अंदर हम आयुर्वेदिक का प्रचार प्रसार भी चल रहा है. तो क्या आप इस बात से सहमत हैं कि इंडिया के अंदर हम घरेलू उपचार जितने जो कर रहे हैं उससे भी डेथ रेट में कमी है? ये 2 चीजें.
सर, आपका पहला क्वेश्चन जो था कि वायरस वैक्सीन कब आएगा, तो आई एम नॉट श्योर, बट मेरा जो स्पेकुलेशन है उस हिसाब से मुझे लगता है कि नेक्स्ट ईयर अप्रैल या मई तक आ जाना चाहिए.
अच्छा, ठीक है.
नेक्स्ट ईयर, अगर सब कुछ सही चलता रहा तो, जैसा कंडीशन अभी है उस हिसाब से.
ठीक है.
और डेथ रेट में डिफरेंस के कई सारे रीजन है. एक रीजन ये है, यूएस में डेथ रेट ज्यादा है क्योंकि यहाँ पे स्मोकर्स बहुत ज्यादा हैं, यहाँ सिगरेट बहुत ज्यादा करते हैं. तो स्मोकिंग की वजह से उनके लंग्स जो हैं ऑलरेडी वल्नरेबल हैं इस इन्फेक्शन के लिए. और ओल्ड एज पॉपुलेशन यूएस में बहुत ज्यादा है कंपेयर टू इंडिया में. यंग पॉपुलेशन ज्यादा है, इस वजह से वहाँ पे रिकवरी रेट ज्यादा है और डेथ रेट कम है. रही बात आयुर्वेदिक की, तो आयुर्वेदिक का प्रचार प्रसार इंडिया में ज्यादा है, बट वो कितना हद तक कारगर है ये मुझे पता नहीं, बट डेफिनेटली आपको वो कम वल्नरेबल बनाएगा टू गेट द इन्फेक्शन. एज आई सेड अर्लियर कि सपोज मेरे थ्रोट में ऑलरेडी इन्फेक्शन है, आई कम अक्रॉस COVID-19 वायरस थ्रू माय नोस्ट्रल, एंड that infected region has a more chance to entrap those COVID-19 viruses and start and proliferate, right? कमल अगर कीचड़ रहेगा बहुत सारा तो जरूर खिलेगा, अगर धरती में लगाएंगे तो नहीं खिलेगा. तो धरती is like a healthy stage, but कीचड़ is like a stage where people are more vulnerable. So that's why I say that do gargle with your salt water every day, so the throat is fine and you are not vulnerable to get any infection.
और जैसे अपने नानी दादी जो है घरेलू उपचार जो बताती थी अपने को, वो घरेलू उपचार भी आज की तारीख में कारगर है. क्योंकि मैं खुद स्वयं के ऊपर पिछले 3 महीने से मैं चाह रहा हूँ कि मैं ऐसा समझता हूँ सभी लोग कर रहे होंगे कि मॉर्निंग में उठते थे हल्दी, गर्म पानी में हल्दी लेना और अपने जो काढ़ा होता है वो रेगुलर पीना, हल्दी दूध पीना. ये चीजें आम हिंदुस्तानी शायद घर घर में रेगुलर हो रहा है, ऐसा मेरा मानना है. तो इस कारण से हो सकता है कि थोड़ी डेथ, ये पेशेंट मिल जरूर रहे हैं पर रिकवर जल्दी हो रहे हैं, मेरा ऐसा मानना है.
सर साइंटिफिक एविडेंस तो हिसाब से नहीं है, बट डेफिनेटली इम्यून सिस्टम हमारा बहुत अच्छा है कंपेयर टू वेस्टर्न कंट्रीज. और वो इम्यून सिस्टम अच्छा इसलिए क्योंकि हम कई सारे एंटीजन से एक्सपोज होते रहते हैं, हमारी बॉडी में ढेर सारे एंटीबॉडीज हैं, और डेफिनेटली हमारा खान पान भी ऐसा है कि हमारा इम्यून सिस्टम थोड़ा बूस्ट होता है इन सब चीजों से. तो डेफिनेटली कहीं ना कहीं वो हेल्प करता है, बट साइंटिफिक एविडेंस इस चीज का नहीं है, तो मैं इसको कमेंट नहीं कर पाऊंगा.
ओके,
यूएस में लाइफ.
हेलो
दवे मैडम.
डॉक्टर राम, मैं ये कह रही थी कि बाय द टाइम द वैक्सीन विल गेट डेवलप्ड in USA many of the people will get the herd immunity, don't you think so?
No, ma'am. Until and unless vaccine is in the market, there is no herd immunity.
Okay. Okay.
Okay, Tiwari sir, go ahead.
Sir, sir, thank you, Ram sir.
Thank you, sir.
जो आज एक मेडिकल आपकी तरफ से जो मेडिकल टर्म्स में जो सारी चीज़ें सब बाहर रख आई, हमारे लगातार दो लास्ट सेशन जो है, एक आपका सेशन और इसके पहले आपके सर का सेशन, उसके कारण बहुत सारे कन्फ्यूजन्स रिगार्डिंग कोविड क्लियर हुए हैं। बहुत बहुत थैंक यू, क्योंकि आपका जो टॉपिक था कि डिट्योर जो एक वर्ड है आपके एक टॉपिक में, एक बहुत लंबा रास्ता हमको बहुत पार करना है, बहुत कठिन रास्ता हम पार कर रहे हैं, इसके आगे का पार करना है। और वे जैसे कि मेरे माननीय प्रधानमंत्री जी ने भी कहा है कि इसके साथ जीने की एक ताकत हमको खड़ी करनी है, और ये भारत ने इसका एग्जाम्पल प्रस्तुत किया है। बहुत बहुत धन्यवाद आपका, आज आप हमारे सेशन में हमारे साथ रहे, बहुत बहुत धन्यवाद। मैं प्रिंसिपल सर से चाहूँगा कि वो आखिर अंत में 2 अपने शब्द कह करके इस सेशन को अंत करें। थैंक यू सर।
डॉक्टर राम सर, हम बड़े गौरवान्वित थे कि आप हमारे कॉलेज में पढ़ाते थे, ये जान के बड़ा अच्छा लगा। और आप प्रोग्रेस करें, बहुत अच्छा सेशन रहा और बहुत जानकारी आपने जो दी वो बहुत वैल्यूबल है सर, सभी पार्टिसिपेंट्स के लिए बहुत बहुत धन्यवाद आपको, थैंक यू वेरी मच। और बीइंग ए पार्ट ऑफ माय फैमिली, कॉलेजिएट फैमिली, आई विल रिमेंबर यू, मैं बार बार आपको याद करूंगा। और बाय वे आप डॉक्टर ज्योति के माध्यम से हम चर्चा में बातचीत करेंगे, और आगे भी जब जरूरत पड़ी हम आपको याद करेंगे।
बिल्कुल सर, बिल्कुल, आपको जब भी कोई चीज लगे आप बिल्कुल बेहिचक बोलिए।
थैंक यू वेरी मच, थैंक यू
सर, थैंक यू।
सारे पार्टिसिपेंट्स को बहुत बहुत धन्यवाद, बहुत बहुत धन्यवाद, थैंक यू।
थैंक यू मैम।
Thank you, sir. Thank you, Dr. Ram.
Thank you.